In this video, we're going to talk about a general overview of what your body does on a cellular level to break down food. To start off, I've got a picture of this delicious looking grilled chicken salad. And although the salad has a lot of different things on it, your body's got one goal for this, and that's to break it down into small components that it can deal with and use those components for energy. There are three main stages in cellular respiration. Now, that term cellular respiration in general refers to all of the ways that a cell is going to make energy. So you'll see times that it is represented as aerobic cellular respiration versus anaerobic cellular respiration. And a lot of times they're looking at different organisms when they do that. We're looking at humans here, and we're kind of examining what we do for the most part. Aerobic means that there is oxygen present. We're going to assume for the sake of this video that cellular respiration is always aerobic, and then anaerobic, which is without oxygen, we're really going to lump it under a term called fermentation. So let's go ahead and talk about cellular respiration. It has three main stages that occur. The first stage is glycolysis. So you'll see it here. We're going to start with the molecule glucose. Now, I need you to understand that cells can use a lot more than just glucose, but we use it as an example to explain it because it's very simple and we know the pathway fairly well. The first thing that's going to happen is we're going to take glucose and glycolysis, break it into two molecules of pyruvic acid, and we're going to get some energy out. This is not a lot of energy compared to what we're eventually going to get. Those two pyruvic acids will then move to the second portion called the Krebs cycle, and notice we went from this exterior portion of a mitochondria into the interior of a mitochondria. Glycolysis happens in the cytoplasm, Krebs cycle, and electron transport chain happen in the mitochondria. So these two pyruvic acids move into a Krebs cycle. We're going to completely break down those pyruvic acids, releasing a little bit of energy, CO2, and a lot of our energy that's left is actually going to be moved on to the electron transport chain in the form of NADH and FADH2. Let's take a look at glycolysis more closely. Glycolysis on its own is an anaerobic process. And what that means is that it doesn't require oxygen. It does take place in the cytoplasm, and no oxygen is needed to have glycolysis happen. And we'll take a look at how it's related to fermentation later. So if we zoom in on glycolysis, the first stage is what we could call an energy investment phase. Because what you're going to do is you're going to take glucose and you're going to add two ATP or two of these energy molecules to it. You might think that's counterproductive, but it's the same idea of how to get a fire started. You got to put a little energy in to get the fire burning. Then the fire is able to release a lot of energy in the form of heat. Breaking down glucose isn't exactly fire, but to get this process rolling inside of the cytoplasm, we got to put some energy in. So glucose will first have an energy investment phase where some energy will be put into it, and then we'll have what we might call an energy payout phase. We're going to get some energy out of it. And you notice there's an intermediate in here. We're not really worried about what the names of those intermediates are. We really want to focus on the major functions. So glucose, you put some energy in, and then you're going to get out. So notice we get 2 ATP put in, and then we're going to get out 4 ATP. That means you are going to net a total of 2 ATP from glucose. You do get 4 coming out, but it's the idea of buying a $2 lottery ticket. If you bought a $2 lottery ticket and you won $4, you didn't actually win $4 because you paid $2 for the ticket. What you actually won was your money back, the $2 you paid, and then $2 more. That's the idea here. Even though we get 4 ATP out, we net 2 ATP. The other thing that we're going to get out of this is NADH, and these will be used in the electron transport chain. And the other very important thing we get out of this are the 2 pyruvic acids that will eventually move on to the Krebs cycle. So when we talk about glycolysis, it requires no oxygen, happens in the cytoplasm. It's got an energy investment phase outlined here in green where we're actually going to put some energy in. Then it's got an energy payout phase where we're going to get our 2 NADHs, 4 ATP, which makes a net of 2 because we already put 2 in, and our pyruvic acids. Glycolysis is very important. It's the first step in cellular respiration as well as fermentation. Some of the advantages of glycolysis are that it produces very fast ATP, and it doesn't require oxygen. Things that we get out of it are 2 ATP, 2 NADHs, and pyruvic acid. The next stages are the Krebs cycle and the electron transport chain. Both of these are what we call aerobic, which means they must have oxygen. And where that actually takes place is at the end of your electron transport chain. The oxygen you breathe becomes the final electron acceptor for some hydrogens. And without those hydrogens and this oxygen there to take in that electron, the electron transport chain backs up. and then it will back up your Krebs cycle and the whole thing shuts down. So basically speaking, the Krebs cycle and electron transport chain are dependent on this oxygen right here for taking in these electrons that we're going to use in the electron transport chain. Without that oxygen, you would not be able to go through the Krebs cycle and electron transport chain. Both of these do happen in the mitochondria, and what they'll eventually yield, besides being CO2, which you breathe out, what they'll eventually yield is way more energy than you got from glucose, from glycolysis. So let's take a look at the Krebs cycle. Basically, what we're going to make is CO2, a little bit of ATP, some NADH, and FADH2s. Now, a lot of these are acronyms. If we think back, ATP is adenosine triphosphate. That's an energy molecule your body can readily use. We're going to generate a little bit of that from the Krebs cycle. However, new to us maybe is NADH and FADH2. Don't worry about what these acronyms stand for, like understanding that ATP is adenosine triphosphate. Just understand that NADH and FADH2 are what we call electron carriers. They kind of act like a bucket full of coal. That bucket is used to carry the coal. It's going to drop the coal off at a furnace. The furnace is actually going to use that coal to produce heat, and the heat's what you want. With NADH and FADH2, they carry these electrons to the electron transport chain. Once they're there, those electrons can be used to make ATP, but we got to get them there. So NADH and FADH2 are electron carriers, and they're going to allow us to get these electrons to where they really need to be. So out of the Krebs cycle, we get some waste in the form of CO2, some energy in the form of ATP, and we still have some stored up energy in NADH and FADH2. If we zoom in and look at that a little bit more closely, you will notice that there are a lot of intermediates that take place in this process. We're not worried about all of those, but we can take a look at the very beginning of it. Pyruvic acid is going to enter the mitochondria, and if you remember, pyruvic acid is made because of glycolysis. We're actually going to oxidize that pyruvic acid, and in that process we're going to turn it into something called acetyl-CoA. But what we get out of it is a little bit of NADH, an electron carrier, and some CO2. So one of our CO2s is taken out of that pyruvic acid. That acetyl-CoA that was pyruvic acid now enters the actual Krebs cycle, and it's going to move through that Krebs cycle. And you notice that it is a circle, just like our Calvin cycle. It ends at the same place it starts. So it's also known as the citric acid cycle because it starts with citric acid. But we're going to release 2 more CO2s, and you'll notice that glucose had 6 CO2s, but we're only going to release 2 for each turn of this process, and then 1 from oxidation. If you remember, glucose is split into 2 pyruvic acids. So there's only 3 CO2s here, and all of these carbon dioxides are going to be waste products. The other things that we're going to generate are all of our NADHs, which are electron carriers, and our FADH2, which is also an electron carrier, and then a little bit of ATP. The totals on the left are per glucose molecule, so they will be doubled from the picture on the right because we're talking about 2 pyruvic acids. But what we'll end up with is 6 CO2, 2 ATP, 8 NADHs, and 2 FADH2. If you're keeping count, we got 2 net ATP from glycolysis, we have 2 more from Krebs cycle. So we've only got 4 ATP of what will potentially be about 36 when it's all said and done. So even though the Krebs cycle is very important, it all on its own doesn't release a lot of usable energy, and we need the electron transport chain for that.